with this catalog that Ed Wilson and others are developing, do we have most of what we need for that catalog? Are we in early stage of discovery? We tried to combine every sequence that we could early on with the Global Ocean Sampling Database, which was twice the size then of all the public data sets, together with all the genes and GenBank and every other database we can find. It took about 1 million CPU hours, and we came up with around 50,000 major gene families thus far on the planet. But any new sample we take from anywhere adds new gene families on in a completely linear fashion. We can break this down a little bit. Those of you who are interested in mammals, there's really no point in sequencing another mammalian genome if your goal is to discover new genes. Understanding new gene content and variants and regulation, it's worth doing, but we've basically saturated the mammalian gene pool. And guess what? We're a very tiny fraction of the gene pool on our planet. When we look at bacteria, archaea, viruses, we're totally in a linear phase of discovery with no hint of tapering off. We published these results last year in a special issue of PLOS Biology, and we've certainly got a lot of compliments from going into a public access journal, people who can now instantly download this information around the world. We extended the expedition after the circumnavigation last year, going from Panama up to Glacier Bay, trying to build into a number of independent sampling cruises off the west coast. We took samples off of melting glaciers. We have them now from various depths, doing time series studies off of La Jolla, et cetera. We plan next year to extend further. We've also had extensive sampling done by one of our scientists, Jeff Hoffman, with multiple trips down to Antarctica, to the various lakes there, and taking samples to and from Antarctica. Next year we plan to go over to the Baltic Sea and then down to the Mediterranean and Black Sea. Many of you know these are totally unique bodies of water, and I think it will greatly extend our gene inventory of the planet. Now, much of our work has been funded by the DOE, but also by the Gordon and Betty Moore Foundation, who, trying to put some context on this data, funded us to do 160 genomes from cultures that people had in their labs around the world. And it turns out many of these are probably going to be from that very long tail of rare isolates in the environment, and they don't really provide much context for this. This is very important, as you'll see as we start to look at the bacteria associated with our own species, that there's so much diversity there that sorting out this long tail is going to really be some challenges. So we've doubled the number of genes twice in the public databases, first in 2004, and then that special issue in PLOS Biology describes 6 million new genes. From our continued sequencing now, our database has about 20 million genes, and as other people pick up this technique, this is going to grow at some phenomenal rate. We have no idea where it's going to start to plateau out. This sort of goes along with what we see of all the genomic data. It's so far beyond what anybody expected to deal with in data, and I'm proud to say we've contributed substantially to that problem. Gordon and Betty Moore Foundation gave us, along with UCSD, a $25 million grant to develop a unique type of database, as an example, called CAMERA. And this database, it's not just a database, it has all kinds of associated information with it, as well as a compute structure. We require people to sign in for this database, and the reasons for this have to do with the Convention on Biological Diversity. Many countries agreed that we could put the data in the public domain only if we tagged it that it came from their country or their waters, and that users of this data would acknowledge the country of origin and the genetic patrimony of where the sample was obtained. But we have satellite data as metadata. We have high-definition video, high-definition photographs, GPS data, chemical data from these sites, and now we're mixing this in with all the data that comes from these different oceanographic cruises, and it's getting to be quite substantive. The other thing, it's about a 5 teraflop computer that any of you can access to do some of these calculations. This exceeds the compute size of what most institutions have, and this is available for free through the internet. There's over 2,100 registered users from 50 different countries. You can see them roughly on this map, and it's growing all the time. Even though it's only been up for a short while, there's a fairly substantial number of publications already from this data set. Now, our initial goal was to apply this to the oceans and to humans, and so there's this metagenomic approach that covers things everywhere. We have 23,000 genes in our 100 trillion cells, but we have another 100 trillion cells associated with us. They're microbial cells. You may want to go home and take a strong shower after watching these slides, but you have about 10 million genes associated with you right now. You have about 1,000 species in your mouth, and as you can see from this chart, your other skin, other cavities are quite well populated as well. We've always known that we've had some microbes associated with us until we started sequencing these populations. Nobody had any real idea. Now there's major studies, and NIH is funding them to look at a lot of human diversity and look how the populations change. What do all these microorganisms do? It's not clear, but the picture gets even more complex when we look at all the chemicals in the bloodstream. Our genome can produce proteins that can make around 2,400 different chemical compounds. So we're not all that diverse in terms of chemistry. If we look in the bloodstream of any of you after you have a meal, we'll find about 500 different chemicals in your bloodstream. That's a lot more than most people imagine. You think if you take an aspirin, you maybe have some aspirin circulating in your blood. Of those 500 chemicals, 60% are from our human metabolism. 30% are from the various species you just consumed, but 10% are from what we feed our bacteria. So think about it. Right now you have about 50 compounds circulating in your bloodstream as a result of bacterial metabolism. We have no idea, do these cause Republicans to be conservative? Do they, we have no idea what these chemicals do. Are they psychoactive? Do they make you depressed? Everybody argues you are what you eat, but you're also what you feed your bacteria. So this opens up a lot more avenues of studies of our own diversity. So can we pare life down to its basic components? Many people have tried a variety of approaches to that. We've been studying this most simple cell, if a cell can be simple. It has around 500 genes, 482 protein coding genes, and 43 RNA encoding ones. And we asked, again, very simple questions. If this cell had roughly 500 and Haemophilus had 1,800 and we have 23,000, was there a simpler set? Could we actually define life at the genetic level by saying, here's a minimal operating system, a minimal gene complement? We started asking questions, how many of these genes are essential for life? What's the smallest number needed? And ultimately, could we design and synthesize the chromosome to help answer those questions? We tried comparing a large variety of genomes. That wasn't very helpful because it turns out the different species don't have that much in common, even closely related ones. We tried knocking out genes, and I'll show you in a minute why that was complicated and why we ended up with the synthetic genome. You probably can't see all these little black triangles on there, but that's every place a transposon would insert in this genome. When it does, it knocks out genes. They're highlighted in these large boxed areas where they're concentrated. But it was a very humbling experiment for a molecular biologist to do because something that was obvious to all of you, we proved the environment was important.